So Ori did his PhD in Israel and then went to do a postdoc at the EMBL as an iPod shared between the groups of Marco Caxon and John Briggs. He then went back to Israel and became a group leader at the Weizmann Institute, where he is still doing his correlative microscopy work. He's a very valuable member of our work group 1 within COMULIS, and I'll hand over to him for his presentation.
Okay. Everything works. You can hear me well, and sort of slides seem okay. Good. So what I thought I would do today, even though sort of I've committed to discuss the high accuracy correlative microscopy paradigm at room temperature, in order to make this more of a conversation and sort of try to deduce what are the governing principles of the decision-making process to do CLEM and which approach to use, I thought that I should first start by telling you from a scientific perspective why I even got into correlative microscopy, what is the reasoning. I'll present a sort of typical cell biology question or challenge that we are facing, I would say, as a research community. And then I'll try to sort of deduce from that which approach to choose and why. And let's see if it makes sense. It's a bit of an experiment. So my lab and me personally, since pretty much forever, I've been interested in membrane remodeling, which is, for a lack of a better statement, is simply essential to life. The very reason we are able to have this conversation today is because at some point a sperm cell fused with an egg cell, for example, and that's a pretty dramatic process for a cell to do during its life cycle. And this is absolutely reminiscent to what we are experiencing now. One sort of membrane compartment transferring genetic information. This in this regard, it's a virus into a host cell, again requiring membrane fusion and remodeling. In this example, for example, in the images, you can simply see a wide field microscopy image of myoblasts, muscle cells, satellite cells that are involved in myogenesis and muscle regeneration. And you can see at this resolution how they start from mononucleated cells and become these elongated fibers. So, you know, a lot of membranes being remodeled. Another event of membrane remodeling, which is very sort of fundamental to cell biology, is vesicular trafficking, both within the cell and between cells to traffic material and information. And in the example, you can see a rather large vesicle. In the beginning of the movie, it's full with cargo, which is fluorescently labeled, and that cargo flows out of the cell. You can also see that upon the fusion of such a vesicle, a protein machinery, a protein that is labeled in red, in this case, assembles on this vesicle and facilitates the cargo release process. And yet another example of membrane remodeling is organelle sculpting, which is essential to maintain organelle structure and function. As examples, one can think about the remodeling, constant remodeling of the mitochondrial networks or the ER into tubes and sheets. And yet in another form of imaging modality, you can see two mitochondria fragments, I should say, with their internal membrane organization, and they are in some sort of close supposition, either undergoing fusion or fission. As well, you can see in this sort of three-dimensional reconstruction some other membrane organelles around that might be associating with these mitochondria. And so if we focus on one archetypal, you know, sort of an archetypical process, I think some of the challenges would emerge. And so let me present you in short, clathrin-mediated endocytosis. Just to say, every cell in our body currently is performing clathrin-mediated endocytosis. This is a process conserved across eukaryotes and in plants, and so it's very fundamental. And what actually happens is a subdomain of the membrane is being reshaped into a cargo transport vesicle. And in order to undergo this transformation, it recruits a protein machinery which I will only introduce in part. Namely, you have cargo receptors on the surface of the cells. These are typically transmembrane proteins that bind soluble agonists, for example, on the external part. And then you have an adapter protein family in the cell that is able to recognize these receptors under certain conditions, for example. And once this happens, this results in the recruitment and concentration of a protein machinery that involves predominantly a very famous molecule that also names the pathway, clathrin. So clathrin is quite a large protein complex. It's a hexamer that assembles into these really gorgeous polygonal coats around the membrane. And another important protein module depicted here in blue is dynamin, which essentially severs the membrane neck connecting the vesicle to the membrane, releasing the vesicle into the cytoplasm. So I would say that for the rest of the row, just remember clathrin as this coat and dynamin as the scission module, but also pay attention that we know of at least 20, if not more, accessory proteins that are recruited to this membrane domain as this process progresses, and defining sort of functions for these proteins, either in concentrating cargo or bending the membrane or regulating the rate and flexibility of this process, allowing it to be adaptable to the various demands of life or as a response to injury, for example. It's extremely difficult to assign these functions. And the reason is that like many processes that happen in the cell, this event is on the order of 100 nanometers, so it occurs way beyond the standard diffraction limit of light. It's highly dynamic, and it's also complex. It involves a complex protein machinery and complex regulation. So even if I'll take sort of a simple fact, we can see if we use electron microscopy, and this is a form of electron microscopy where we only look at the basal surface of the cell and we sort of coat it with a metal so we can have high contrast in this imaging modality. You can see clathrin in sort of two forms. One is these flat sheets, and the other is in these curved baskets, and we can get exquisite resolution of these curved baskets by cryo-electron microscopy, for example. However, understanding whether this flat sheet is the precursor for this vesicle, or are these completely different species, is not trivial to determine. And let me try to sort of take you through the thinking of why that might be and connect it to the need for correlative microscopy. So endocytosis can be imaged by various modalities. A very common one that is now, I would say, available almost everywhere is confocal microscopy. So confocal microscopy allows us to look at a sort of slice through a three-dimensional object. These objects can be quite thick, like a tissue. And this optical slicing allows us to obtain quite nice resolution in 3D, however, not sufficient to resolve with high spatial resolution the process of trafficking. Common issues with this is that once a vesicle would pinch off the membrane, it has a huge volume to move through, so we are unable to track the migration of vesicles easily through this gigantic volume. So, and the other challenge is because we are optically slicing, meaning we have to spend time going through this very thick object, we lose on temporal resolution. And since, and I didn't mention this, endocytosis occurs on a time scale that is between, I would say, 20 seconds to 2 minutes. It's quite a rapid process. We need to be imaging at very high rates if we want to capture this process within a large volume. Now, it's possible to overcome some of the challenges here if we reduce the dimensionality. For example, if we were to use total internal reflection fluorescence microscopy, if you are unfamiliar and just keeping sort of basic terms, this is an imaging modality in which we only illuminate about 100 nanometers at the basal surface of the cell. The principle is that the laser beam is totally internally reflected once it hits the interface between the glass, the substrate on which we grow the cells, and the liquid. And so we illuminate a very thin slice, and so we bleach less. We can image very rapidly because we are not slicing through the object. So in a way, this is a reasonable compromise. We relinquish sort of the three-dimensionality, and what we get is in sensitivity. So we can resolve these sites very nicely. We can gain very good temporal information. And the way we sort of know that the vesicle has formed is that when it pinches off the membrane, it moves out of the field that is illuminated and disappears from the field of view. So by just reducing the dimensionality, we gain a lot in temporal resolution and in spatial resolution, but it's not nearly enough, because if we zoom into the object, it's still completely not visible. We cannot see what is going on at the membrane level. Now, TURF can be enhanced if you have access to more advanced microscope with structural illumination. Here is an example from already a few years back. So still we are looking at a two-dimensional image, but now if you look really closely at the image, you can see that we can see the little hole. So now imagine that the vesicle is forming into the plane, and you're sort of seeing the cavity of that membrane being formed. So some spatial resolution is there, but it's two-dimensional. And now, in order to increase this even farther, and I think I've noticed Jonas is in the audience, so this is for you. If we want to enhance this even farther, we sort of give up on the temporal dimension. We fix the cell, so we don't have to worry about things moving around while imaging. And then applying now, for example, a TURF storm paradigm, we can even gain three-dimensional information, in this case about the architecture or the ultrastructure of this clathrin coat, this coat that builds around the vesicle. Now that we have given up on temporal resolution, we can move on to electron microscopy resolution. So for example, by transmission electron microscopy, we can get already the shape of the membrane and the coat. So I would say this is a more comprehensive view of the object, while in fluorescence microscopy, we can only see what we have labeled. In electron microscopy, we also see what is not labeled. So that's an additional advantage going along this trajectory. And finally, if we go in the direction of cryo, electron microscopy and tomography, while the compromise here, I would say, is on the sort of contrast that one would get, because we don't use any staining whatsoever, all the structural information in the sample is intact, and by using averaging, we can even obtain sort of structural information about the organization. Now, given that our goal maybe, and I hope you would agree, is at the end, sort of, if there is one, to understand all biology at the level of proteins and even lipids moving around, to understand their movement, their conformational changes, and how these changes result in biological function, I would say, imaging is extremely appealing as an approach, but there are some challenges. We can use different modalities to get different levels of information. However, this results in this kind of compromise, that the more we want or require spatial resolution, we have to give up on the temporal resolution. And this is not the only challenge that I sort of try to allude to with this slide. There is also, not always, but in most cases, the transition from looking at the sort of the tissue or the cell at its native condition. As we require higher and higher resolution, we normally reduce the dimensionality, and therefore we might even move completely into an in vitro paradigm to gain more spatial resolution. And another important consideration is the throughput one can obtain, because to address biological questions, one has to visualize the event over and over in multiple different days, etc., and to come up with sort of a statistical representation of reality. And therefore, while light microscopy methodologies tend to have reasonable and even high throughput, the more we have a demand for high spatial resolution, there is a compromise for the throughput that one can obtain. And what we try to do with correlative microscopy, and I would say these are the examples where you gain the most out of doing that, is we try to make a better compromise between these different elements to apply one, two or more imaging modalities to the exact same substrate and make a better sort of, and find ourselves somewhere in the middle. So to try and summarize what I've just told you up to this point is that while it's very clear that fluorescence microscopy enjoys and benefits as a strength from molecular specificity, we know molecules that we have labeled where they are in the cells, and everything else is black, so we can really target those molecules. And it also contains the rich dynamic information that we want. It is limited in resolution, and on the other hand, you have electron microscopy that gives you a much better or a more comprehensive view of the environment. Also, unlabeled things are visible and high resolution. And we have to balance these two things out, but there are more challenges to be balanced. So the more if we want to be on the absolute high temporal resolution, we would have to reduce the volume, we would lose on spatial resolution, and we would also have to probably already sort of thin the samples, and it will be of lower throughput. So this is this kind of idea that no matter what edge of this pentagon you will go to, if you want a large volume, you will have lower spatial resolution, lower temporal resolution, etc. And we try to make this compromise. So assuming that at this point you agree and you have a fantastic question to ask using that correlative microscopy would be ideal to address, you normally come upon a big challenge or issue, and that is that there are many different imaging modalities in both of these paradigms. The fluorescence or light microscopy is too much. So this gives normally students, at least my students, this reaction. They realize that they're left alone at home, so they're happy, and immediately once they realize the meaning of that, they panic because there is too much information, if you will. And as a community, and I haven't sort of said it in the beginning, but as a member of COMULIS, I really benefited a lot in refining my thinking about CLEM, because as a community, until now, we have created a lot of such diagrams. So we can really detail all of these different workflows as flowcharts, starting from fixation, etc., going down different avenues. But in reality, this is not so helpful when you want to start, because how do you, apart from understanding where you are going from your live sample to the EM modalities, what is the compromises that you will be making along the way, and what information you will have intact is not really easy to understand. So instead, I want to try and offer a sort of simplified view of this. And that is by starting with a very simple question: what is it that you need to see? And the reason is because if you need to see a cell or an organelle, which I would say is up to, let's say, 500 nanometer, or a vesicle or a virus that are on the order of 100 nanometer, this already defines a lot about the type of methodologies that you will benefit from. Firstly, it defines whether you are at a resolution of the scanning electron microscope or the transmission electron microscope. Because, for example, a 100 nanometer vesicle is extremely, if not, let's say it's possible to resolve it, but to get quantitative or useful information in scanning electron microscopy is not so trivial. So you already would expect that if I want to look at my vesicles, I would be sort of recruiting a transmission electron microscopy paradigm. The other thing that that understanding what are the objects that you want to correlate is that this very much defines the correlation precision that you need to obtain. And that correlation precision is defined very much by when along this very sort of complex multi-track trajectory you would go. Namely, let's say you just wanted to know something about a cell where you have expressed some silencing the RNA or a knockout, and you know that that cell is knocked out by some fluorescent marker that you've put in. You only need to identify that it's a cell. In this case, it doesn't really matter for you when you image your sample. It can be imaged before you prepare the samples for electron microscopy, and you'll be able to find back the cell of interest by using very global features of the sample. For example, a grid that you have on your substrate. So this, I would say, is very sort of requires low precision correlation. However, as you go down these different objects that are becoming smaller and smaller in a sense, you will need to have higher correlation procedures. So let's take, for example, this idea of looking at endocytosis. If I were to image my cells live, then by the time after I've finished imaging the process and I wanted to fix them for EM, because I hope at this point we are all on the same page in terms that there is, that electron microscopy has no dynamic information. It's all a post-fixation method. In that time that it took the fixative to do its job, whether chemical or cryogenic, your object continued to move. So it's no longer, physically no longer necessarily in the position that you imaged it, and therefore your ability to correlate it is diminished. Also, if you are imaging a cell which is about, let's say, 10 micrometer in thickness, and then trying to identify the same object later on in relatively thin slices, because again, a transmission, for example, electron microscopy is restricted in the penetration depth. The electrons have to pass through the sample. You are again confounded by the fact that finding back at which of the optical slices your object resides can be extremely difficult and time consuming. So if to sort of summarize what are the consequences of imaging live, correlation precision is diminished. The throughput of the CLEM procedure is slightly diminished because you'll be spending more time looking through the different sections. However, you have imaged your fluorescent signal when your cells were as close to the native state as I would say, if I were to define the native state, I would say this is the cells currently, working in my body. I remove them to the dish. I've already slightly removed them from their native state, but it's as close as let's say we can currently get. Moving down the line, if we image the sample post-fixation, either, this is not important whether it was a fixation by cryo or chemical at this point, although there are considerations here, definitely, which I will touch upon, then the sample is not moving anymore. You already gain a lot in correlation precision. However, your sample is still thick. You're still looking at whole intact cells most of the time, if not tissues. And therefore, there is still the issue that you will need to align between the optical slice and the TEM or SEM slice. So this, I would say, is quite suitable for organelles, but can be quite challenging if you want to find something as small as a virus, not to mention a protein complex, which is, of course, an additional option. Finally, we can also image after the cells have been fully prepared for electron microscopy. Some common examples that you may be familiar with, after we have milled a thin lamella under cryo conditions, or after we have sectioned under cryo conditions or in resin, or whether we have made thin ribbons for SEM analysis, or a block for SEM or FIB-SEM analysis. Irrespective, the sample is already at its final state. It has undergone through this entire process, and therefore correlation precision will be the highest. Okay, so far I hope it's all good. Normally, I would be in an auditorium and can see if you are falling asleep, but here I will have to sort of assume there is a live audience out there. Okay, so how does this relate to what I told you before? Let's take the example of endocytosis. I've already shown you this cell. It's imaged by a TIRF, by total internal reflection fluorescence microscopy, with two markers that I hope you remember from the beginning. Clathrin, which will make this coat, and dynamin, which will remove the molecule, the vesicle from the target membrane, or from the origin membrane, sorry. And this is how it looks in a chymograph. So now I've simply plotted time on the x-axis, and you can see a clathrin arriving at the membrane, accumulating there, and then staying, And at some point, dynamin is recruited in a pulse, and the two signals disappear from the membrane, presumably because a vesicle has pinched off. So it really sort of mimics, it has the feeling of this dynamic process of endocytosis. But of course, we cannot relate, we cannot say, for example, when is dynamin actually recruited along this trajectory directly from the imaging. Now, because these cells have endogenous labels, meaning they express RFP and GFP from the endogenous locus, and the expression levels are as close as one could try to get to the endogenous expression levels, the signal is quite dim. So I would say that because I know my fluorescent signal, I can already confidently say that I have to go through cryoimmobilization in order to maintain my fluorescent signal, irrespective of whether I image now after I have immobilized only, or if I proceed with the processing of the sample. Because at the end, cryoimmobilization, since we know it is also very useful for preserving protein structure, it is also preserving the fluorophore structure. Because I need very high correlation precision, my object is 100 nanometers. Then I want to image post-processing. I want the samples to really be at their final state. And therefore, the paradigm that I'm using here is after cryoimmobilization through a process of free substitution. The samples have been embedded already in resin. And these tend to be special, let's say, resins that perturb or make as little interactions with the sample as possible as to not perturb protein function. And so what you are now seeing on the right is just a slice, a 300 nanometer slice through these cells that express the clathrin and the dynamin markers. The very bright multispectral dots are actually beads that we add onto this section. And this is because in order to have any sort of correlative modality, you need to have common features between the two samples in order to be able to align the information one onto the other. If you do not require very high correlation precision, one could simply use objects, like for example, phallopodia, the outlines of the cell, anything that is visible in the two imaging modalities can be used. However, if the beads both give quite good correlation precision, and they're also quite unbiased in the sense that they are not part of the sample being examined. And the other thing that is readily apparent is these clathrin puncta. And if I'll zoom in, you'll also see a spot here that also has the dynamin signal co-localized. Let me see if I can highlight it. I'm using a new famous sort of flashy pointer. So you see that there. So this allows me to take images at the very same spot, and I'd like to name some advantages. Firstly, because I have the fluorescence already, the fluorescence map, and I can relocate to the same regions, I can now target the objects I'm looking for. I'm no longer sort of randomly looking across this membrane. And I should mention there are two cells here. There is a cell on the right, and there is a cell on the left, and in between them, you can see the intercellular space. And so instead of sort of randomly looking through this section until I find objects that might be of interest, I am targeting these objects, and therefore I increase my throughput quite a bit. So I have reasonable throughput, I have good spatial resolution, I can see the membrane, and I can visualize the fluorophores still where they are, so with a very high precision. So I would say that you see how this makes sense within this paradigm, that if I want, because I am trying to look at something extremely small, I am sort of locked into this post-preparation paradigm. And so it immediately sort of, I can conclude that without looking at other approaches. What other informations one gets from this approach? Because I can take relatively thick, 300, 500, potentially with STEM tomography up to a micron, without a huge compromise on spatial resolution, I can also do electron tomography and reconstruct a 3D structure. So from this approach, one can get a better representation of the 3D architecture of the object. And the fluorescent information tells me that in this side, there is, with high probability, one of the fluorophores. And in this side, there are two of them. And if you remember the chymograph I showed you, this makes total sense. This is sort of a U-shaped invagination of the membrane. And you see that the other object already has this curvature at the base, meaning this constriction that will eventually undergo fission, releasing the vesicle into the cytoplasm. So the fluorescent information here gave me both molecular composition, but also time. So while we might think that correlative microscopy, simply stated, would give us the dynamic information and the high resolution information together, very often the fluorescent signal or combination of fluorescent signal gives us actually a surrogate for time. Because we know how the fluorescent signals behave one with respect to another, we can use those to deduce at which times these different objects are relative to one another. And this is sort of maybe a less spoken truth about, or realization about, what we do. The only method that gives you real dynamic information is, were you able to image the process live? However, even then, you only get information about the history of the event up to the point of fixation. What additional information one might get from high accuracy, apart from being able to sort of measure the ultrastructure of the membrane and potentially the protein coat, because we have better than 100 nanometer of precision, and the object is on the order of 100 nanometers of more, one can also ask probabilistically, how are the different protein complexes oriented, one with respect to the other? What do I mean probabilistically? One can take the entire data set and ask basically, what is the probability that the signals overlap? What is the probability that they are positioned, for example, at the base? So what you're seeing here at the center of this circle is the maximum probability. So since this is the depiction of sort of the average coat, the maximum probability, or the center of mass of that coat, is here. This gives you a more or less representation of the error that we have, because the real center of mass is somewhere here, and it's shifted. And there is a 10-fold decrease in probability the more you move out to the circle. The same here, dynamine is more shifted to the base of the invagination. And this is again consistent with what we expected, and what, biochemistry and immunogold labeling, for example, as a complementary approach, has told us over the year. Dynamine assembles at the base of the invagination, and clathrin assembles along the bud. So I would say this is a nice confirmation. However, I would also mention that it's quite rare for us to find scenarios in which you really need that high correlation precision. This is unique when your object is firstly not symmetric, like an invaginating segment on the membrane, and also that it's of the right dimensions. Okay, so coming back to this, let's try to analyze sort of together what are the compromises of this method relative to other methods. Okay, so this would look like this. So I would say that since the sample is fixed, right, to begin with, the temporal resolution is not the maximum. It's not so. It also is not the lowest, because we still get some temporal information from using different combination of fluorescent markers. So we know this is very likely an earlier object than this. Spatial resolution can get slightly higher by using even more advanced electron microscopy approaches. So I would sort of place it here. In terms of the native state, we've cryo-immobilized, so that's already good, but then we impregnated the cells with resin. So I would sort of assume we're slightly moving away from the absolute best. The throughput, also quite high because we are targeting, but the volume is only 300 nanometers. So we are really looking at very restricted areas of the cell membrane and the cytoplasm. A very similar approach is the approach combining the method I really started with, this shadow replica electron microscopy. In this approach, the cells are grown on the substrate, just like the approach I just described. However, right after fixation, the cells are unroofed to expose the surface that is adhered to the substrate. And then the nice thing is one can has access to these sites on the membrane, so you can stain with antibodies. Antibodies are attached to fluorophores that are more resistant to the conditions of both imaging and sample prep, so you get much higher brightness and photon yield, I would say, from such preparations. However, you again only have a sort of a slightly better than two-dimensional representation because the sample has been reduced to almost 2 dimensions. And also because the sample is stained with a metal coating, structural information is actually less accessible because what you are seeing is sort of your molecule wearing a big fur coat. So there is a limit. So let's try to put it at the axis, and my apologies if there are slight inaccuracies. This I would not try to sell you as a perfectly objective process, but I would say that temporal resolution is more or less comparable between these approaches, maybe because of the two-dimensionality, but there one could argue the spatial resolution is a little bit less. One could argue that it's a little bit more because we now can use fluorescence microscopy paradigms that give better spatial resolution, like super resolution, storm, etc. The native state, I would say, is a bit less because the cells have been unroofed, the cytoplasm has been removed, so the sample undergoes many washes, and these changes are hard to account for. Throughput is relatively equal. Again, one can look in the fluorescence microscope at huge fields of view, see the locations where endocytic sites occur, endocytosis is occurring, and then acquire these sites with the electron microscope. And in terms of the volume, it's again slightly less, as I mentioned. Finally, here is a completely different modality that is sort of not covered by correlative by CLEM because it's atomic force microscopy, but still yet another high resolution approach that is sort of intermediate between light and electron microscopy. And the nice thing is that you see one can image even live, so you can in parallel acquire the fluorescence and the electron microscopy information, the AFM information, albeit it's two-dimensional, as in turf microscopy. If one wants better resolution to really harness the high resolution of the AFM, the sample has to be fixed, so the temporal resolution is gone, and then the resolution is actually quite nice. Let me try and use my fancy pointer to zoom in a little bit. You see, you can actually resolve the architecture of the coat and potentially assign it to different stages, still by using different fluorophores. So again, just to compare these methods, while the volume here is two-dimensional completely, even I would say you get less of a three-dimensional representation than with the replicas. Throughput, though, is much higher. If you image live, it's way closer to the native state. The cells sort of are alive and kicking, but you lose a little bit on spatial resolution, and you may gain something in temporal resolution. Maybe yes, maybe no. That's debatable. Okay, so I am not sure how I'm doing with time. So I can actually conclude here. Paul, what is the status?
I think you have another 5 to 10 minutes, if you want.
Yeah, sure. Let's say I just wanted to use the opportunity to point out that in terms of a workflow, the workflow I just presented, wherein you immobilize your sample, you then using free substitution, embed it in resin, which adds a lot to your throughput because the sample becomes mechanically stable. You can section through it relatively easily. You can, work on it at different days, it's less sensitive to environmental perturbations. There is nothing very different about applying this approach under cryo conditions. What you would do is simply omit the resin embedding step. However, now it requires a more specialized microscope. It might not be the microscope that you have readily available to you. And in this case, in the example, you are seeing an electron microscopy grid, which if you're an electron microscopist, you're used to seeing. And the green blobs are cells that were grown on this grid. And we can easily acquire the same field of view in the electron microscope, zoom in on these same cells, and in this paradigm, again, use beads to correlate the two imaging with high precision, and then hone in on sort of objects of interest. Sorry, this is here, for example. So, and then if we are looking at thick cells, we are either restricted to the thin areas where they grow to thicknesses which are compatible with the electron microscope, or we would have to move to an approach that includes thinning the section even farther. With this kind of imaging, though, in the cell, relatively intact, but restricted to the thin areas, you get sufficient resolution to resolve, for example, the clathrin coat in more detail. And that can be used for further averaging, but it can also be used just to deduce the different architectures of the coat. So I would point out with this, just so it's not completely sort of left hanging, that at the beginning I asked, how can we know that the object is maturing from something flat to something more curved? One way to do that is to now have a good structural representations of the intermediates. And by having both the final objects and now all the intermediates along the way, which you can also sort of bin according to different fluorescent signals, it's in a way becomes only a question of time and effort, and not a question of whether we are able or unable to do it. Okay, so I think here is a good place to stop. I didn't go through the technical stages of the nitty-gritty of how you might do this type of in situ high accuracy CLEM. This is covered quite well already in a book chapter by Wanda Kukulski, who also pioneered this approach. And if you scan this QR code, I hope it's called the QR code, I'm so old, you will be taken to a YouTube video that was beautifully made by the EMBL core facility, which also shows the practicalities of doing high accuracy room temperature CLEM. So with that, I only would like to thank members of my group, in particular in this context to Nadav Sheo, who contributed some of these beautiful slides. Nadav also summarized this conceptual way of choosing the CLEM technique in a book chapter, which we called 50 Shades of CLEM. And that would be, I guess, my inappropriate comment of the day. And with that, I thank you for listening, and I would love to take questions. I guess I should look at the chat, Paul, or what's
Excellent. Thank you very much, Ori. Yeah, I was muted, so that's why you didn't hear me. That is, well, it's not inappropriate.
It's not inappropriate anymore.
Share your screen, then we can see all the.
Oh, yes.
Excellent. Then we can have some questions. I see Joy has put in a question. Joy, do you want to turn on your camera and unmute to ask the question?
I'm struggling to find where I unshare now. Where did this little...
You are unshared, Ori. No worries.
I'm unshared. Thank you.
Or maybe someone did that for you. Joy, are you there, or shall I read out the question?
Sorry, I'm on a phone, so maybe it's better if you read. Thank you.
Yep. Or Ori, can you read the question?
Yes, I can read it. So Joy is asking, what would be time range between your room temperature fluorescence imaging and cryofixation for mammalian cells. I'm wondering how confident we could be that the molecules' locations we determined through super resolution fluorescence imaging which already takes quite long minutes, would still be valid after cryofixation. Okay. So let me see if I understood. So one part of the question is to understand what is the time frame if we were to image the cells live, how quickly can we cryo-immobilize them? So first, just let me clarify, in the main paradigm that I presented, the cells were fixed before I ever imaged them, meaning I already have learned a lot about the fluorescence and the behavior of clathrin versus dynamine, and I use that as a surrogate for time later on. Otherwise, generally, I would say, if your process is not too fast, you can probably live with fixing. The fastest I could do it, and I'm by far not the best model human, was about 20 seconds or so between, grabbing it from the microscope, putting it on the slide and high pressure freezing it. Confidently and reproducibly. I've also managed to do slightly better and slightly worse. So if you were to do that, you need a process that takes longer than that, where you can be confident. Now, if your sample is fixed already, then indeed you can use stochastic super resolution techniques where you need to image over long periods of time because your object is not moving. What that might do to the preservation of your sample later on, that's a question for the experts. So something that I maybe have not said is a shout out to all the magnificent facility staff that are definitely part of Euro-BioImaging, but also part of the audience, because whatever paradigm you choose, you are going to need help. You're going to need experts in the two imaging modalities to sort of guide you through what you might not know. But definitely, I think that if you cryo-immobilized and now you are doing super resolution and your sample doesn't look like it's been cooked in the electron microscope, you can be reasonably confident that it represents sort of the ground truth, the actual native or semi-native state.
Right. Thank you very much.
Excellent, Ori. It might be good if you briefly discuss how long you've been doing the live imaging of this process before you even started attempting the CLEM part.
Well, so in the personal paradigm that I presented here, but actually almost in anything that we take in the direction of high accuracy CLEM, we know already from probably years of study the behavior of the fluorescent molecules. I mean, you do not want to make all this effort to find out that actually, and occasionally we do. Occasionally we look at things that people have characterized and discussed for years. And then when we look under the hood, under the fluorescent signal, we realize it's either not what people believed it to be, or actually it introduces a phenotype compared to the unlabeled sample. So this is something that I would say is actually good, serves science. But normally we want to know quite a bit. So actually, the more information you can harness from studying your sample in the fluorescent microscope, the better. For example, if you want to study a rare event, which only occurs in 10% of your cells, then you are going to need an intermediate stage of sort of mapping all the cells in your sample. While if you are using a sample like I was using, where all the cells, 100% of the cells were expressing the fluorescent signal and had almost identical behaviors in the population, because they've been genome edited, and so they all came from a single cell relatively recently in their sort of culture evolution in culture trajectory, then I did not care. I could look at any slice through any cell, and so my throughput again was quite high. But if I now have to look for that one cell in the whole population that has your fluorescent signal, then one has to rethink where along this process. Maybe because you can indeed image at every stage when the sample is live, before, right after it's been fixed, and after it's been prepared, does not mean you cannot do all of them one after the other in order to get these different levels of information. So one, to try and sort of answer your question holistically, I would say not how long I actually already started working on it after somebody has completely characterized the lifetimes of clathrin and dynamine in these cells way before I even started. So, you know, and we very often get it that people say, okay, I am going to fluorescently label my target protein, and then I am going to characterize it, and then I'm going to do high accuracy CLEM. And I often say, well, one step at a time. It might be that just looking and characterizing the fluorescence is going to occupy and is going to be extremely informative already.
Excellent. Thank you very much, Ori. Any other questions? If there are any other questions, just turn on your camera and raise your hand so I can see you.
Okay, so I have, I think, one question that is private, but it's not private, it's just sent privately. And so, yeah, so that's actually a very good one from somebody I know. So the question is whether one could, I'm rephrasing it a little bit, sorry, Leah, whether one could indeed even increase the throughput higher by somehow automating, for example, the recognition of the fluorescent signal. Could the fluorescent signal be automatically segmented, and then the two maps can be automatically aligned? And so, you know, the whole procedure can be pushed even farther, and the answer is absolutely yes. And actually, EMBL has been instrumental in developing some of the tools, and they are published and available, and you should look into them. And, you know, you should write application to go to EMBL with COMULIS and Euro-BioImaging to learn these techniques. And if we were to use something for these initial alignments, normally it's gradual. This is part of the question. I'm not sort of just elaborating. So I showed the fiducials that we use for the high accuracy correlation, right? This is the very end of the process. Even before then, I had to hone in on my cell of interest, right? And so because in my paradigm, I've already laid the section down on a grid, I have the grid bars as sort of a grid that I can navigate in. So I can hone in on the specific grid squares that I have imaged. And in fact, one of the nicest and easiest ways to use automation in this case is using these grid bars, very thick, very contrasted objects to already align, because you know you have a square in the fluorescent microscope, a field of view, and you have a square in your EM, and they are defined actually by these bars on the grid, and then you only have to solve the rotation. And that does not require any special fiducial. It just requires sort of low frequency information in the image. And then once you've locked into your cell of interest, you can even, with enough precision, acquire the object, because you still have a reasonable field of view. And then if you want to know within that field of view where exactly your vesicle is in a sea of vesicles, then you have to rely on high accuracy. And then you run into scenarios where you may even have to improve your fluorescence microscopy paradigm to resolve things better, even in the EM. I hope I answered. How long?
Thanks.
Yeah.
Yeah.
You also ask how long does it take, but that's a tricky question.
On the fly at the microscope, I know that it's not so easy. You can do the alignment, but then if you have a lot of positions, it takes time to really align each of the images, and the microscope time is costly. So you don't want to spend like more than a day to set up, because if you run overnight, you have like a short time for acquiring. So if you have like, let's say in tomography, like 30 position, it's already quite challenging for a night sometimes.
Right.
And if you spend the whole day doing this, plus setting up the microscope, you can have a loss of throughput doing this at the end.
Sure. So what we did actually, in order to exactly deal with this, that we do one imaging session just for mapping. So you spend the day on the microscope, taking the same region where your fluorescence microscopy images are, Then you let them run even overnight, can be 30, can be 100 regions, very accurately sort of correlated. And then when you go back to the EM, since your section hasn't changed, you use, your EM maps against your, not now maps, but actual, where the position is, to relock on those positions. So then it's very quick. You have a very low failure rate. So yes, these are kinds of flexibilities that one has to think about exactly to get to the throughput that you need. It also, of course, depends a lot on the computers you have, and we don't always have flexibility even interfacing with the microscope as much as we want. So definitely doing some things offline and then to speed up the online is a good idea.
Thank you.
Excellent. Thank you very much, Ori.
Yeah.
Any
Pleasure.
other questions? If not, thank you all for your attention and joining the seminar. Of course, don't forget to sign up to Euro-BioImaging, to COMULIS. Don't forget to download the annual report. I've just done that while Ori was talking, so looking forward to that. And of course, I hope to see you all in about a month with the next seminar of joint Euro-BioImaging and COMULIS.